Okay, good afternoon, everyone. I'm Jishnu. I'm an assistant professor at the University of Pittsburgh. I'm a computational systems immunologist, and we use machine learning and network approaches to study immunomodulation in a wide range of contexts, from infectious disease to autoimmune diseases to autoimmune disorders. And at ISMB, I'm talking today about this method that we have for learning the language of protein and peptide interactions. I'm also giving a talk on GRNs tomorrow in case you're interested, and my student just presented at the CSI COSI, which I'm co-chairing. So today I'll talk to you about interaction language models to decode protein and peptide interactions. Now, protein language models, as this community knows, has become extremely powerful and extremely widely used to solve a range of tasks. But what I'll try to convince you today of is that as powerful and as useful PLMs are, passively using PLM-derived embeddings does not capture all the facets or all the nuances of interactions. And you need a biologically inspired language model to really encode these interactions, which we call an interaction language model. And this was work done in conjunction with my colleague and collaborator, Alok, and co-led by three awesome graduate students in my group, Alyssa, Jane, and Prabal. So we developed this method. This is a representation learning approach called swing sliding window interaction grammar to learn the language of protein and peptide interactions. So this is not a large language model. It's really a doc2vec model. So it's a small but biologically inspired language model. I'll talk through what exactly we are doing. So you could have a short peptide and a full-length protein, or a full-length protein and a full-length protein, and I'll show you examples of both. But what we are doing is we are sliding the short peptide against the full-length protein, and then for these pairs across these sliding windows, the size of the sliding window, etc., these are all tunable hyperparameters, we are using a biochemical metric, in this case polarity, but it could be a different biochemical metric, to encode the properties of the amino acids. And we are computing this number, which is the difference in the biochemical metric. That again can be tuned how to represent this. For the tasks that we have used, the difference is the best encoding. So we now come up with this delta biochemical metric, and using a doc2vec model, we embed this. And this is how we represent the interaction. And I'll show you how this is outperforming passive uses of PLM embeddings. This is in press now at Nature Methods. So we have this very simple architecture which captures the actual language of an interaction. It is not passively using PLM embedding. So you're not embedding the individual proteins and concatenating them, or concatenating the sequences and embedding. You are actually trying to encode the interaction. And we use this for a range of downstream cases. The first being looking at peptide MHC binding. As immunologists, this is a very well-studied and important problem. This is obviously critical for downstream TCR signaling. Now, peptide MHC modeling, there are many methods, dime a dozen methods to predict this. But most methods, what they do is they take all except one or two alleles and then predict on the held out alleles. We said if this is really generalizable, we should be able to learn from a small number of alleles. So for class 1 PMHC interactions, we trained the model using only 8 human alleles. And we were able, both in a cross-validation and in a cross-prediction setting, for completely unseen alleles, we were able to do well. Okay, sure. Now we move to class 2 peptide MHC interactions. Again, using only 5 alleles, we were able to generalize both in a cross-validation setting and in a cross-prediction setting. So far, so good, but there are many methods to do this. So we now started to increase the difficulty level. The first question that we asked was, are we learning biologically meaningful relationships? And for this, we did an ablation experiment where we keep on deleting the peptides. For class 2 PMHC binding, the length of the peptide is between 12 to 25 amino acids, but the core binding motif is around 10 amino acids. This is well known. Some methods explicitly encode this. We did not. But we clearly see a drop in model performance only after we drop below 10 amino acids. Still then, there is almost no reduction in model performance, meaning that we are learning the core sequence de novo. Then we said, let's make the difficulty level even more difficult. So we said, now can we move from class 1 to class 2 PMHC binding? And this no existing method has done to the best of our knowledge, because structurally and sterically these are completely different. Class 1 is capped at one end. The peptide lengths are much shorter, 8 to 12 amino acids. Class 2 is open at both ends with peptide lengths 12 to 25, mostly in the 14 to 25 range. However, when we trained swing on class 1 PMHC binding, we were able to successfully predict for class 2 PMHC binding. So to recap, we do as well as state-of-the-art methods for class 2 PMHC binding, but we can generalize learning only from class 1 to class 2. So this was very interesting. Now we said, let's make it even more difficult, because the real goal is to predict for rare disease-causing HLA alleles. So we trained our model only using publicly available human immunopeptidomics data, but we can predict for murine alleles of lupus and type 1 diabetes. And we have 3 models here in blue, pink, and orange. They are swing models trained only using class 1, only using class 2, or mixed. It doesn't really matter. You can generalize very well from human to mouse. Competing methods, NetMHC PAN, MixMHC2 PRED in green and gray, don't do well at all, and there is a clear length bias. They only do well for average length peptides. We do well across the length spectrum. So now we said, let's look at a different problem. And while modern variant effect predictors like alpha missense or ESM1B do extremely well at predicting pathogenicity, they cannot predict the impact of missense mutations on specific protein-protein interactions. So we biochemically know that the wild-type proteins A and B interact. You only change one letter in A. The missense mutation may or may not disrupt the interaction. Can we predict this? And there are experimental gold standard data sets, both for Mendelian mutations as well as population variants across the allele frequency spectrum. And we do much well compared to state-of-the-art methods, including ESM1B, alpha missense, and EVE. So now for the final part, I said I'll compare this directly to protein language models. So we came up with 4 conceptual paradigms. One is you can just take a foundation model, PLM embeddings. You can also biochemically inform these PLM embeddings, but you are really not encoding the interactions. You can concatenate the 2 sequences and embed, or you can embed and then add the vectors. You can do all of these, and these are the first 2 categories. The next 2 categories are really ILM encodings and then ILM embeddings. What we are doing, we are representing the interaction. And for single context prediction tasks, swing does as well as these clever passive uses of PLM embeddings. I'm showing you the missense mutation prediction task here, but we also did this for peptide MHC. But when we now go to extrapolating across contexts, in this case, going from class 1 to class 2, the only method that has meaningful signal is swing. So the ILM, not the other approaches, have meaningful signal. Again, when you try to generalize or extrapolate from human to mouse, of all of these architectures, the only one that has meaningful signal is swing. So passively using PLM embeddings is very good for single context tasks, but when you're really trying to extrapolate, you need this biologically inspired model. So in summary, swing is a first-in-class ILM that learns the language of protein-peptide interactions across contexts using only sequence. Of course, we can infuse in structural details, but we wanted to generalize and keep it to sequence only. It's a few-shot model to accurately predict PMHC binding across HLA classes and species, can make accurate de novo predictions, learns biologically meaningful information with no prior knowledge, can predict the impact of missense variants on specific protein interactions using sequence alone, and it outperforms passive uses of PLM embeddings and related architectures. And of course, this is my lab of about 25 people. Very fortunate to work with an amazing group of people, and I would be remiss if I did not thank my funders and also the organizers for letting me speak, and to you for your attention. Thank you very much.
Thank you. Do we have questions? No, so we can, in the interest of time, also move to the next talk.
Thank you.
Thank you. So